The enzyme-linked immunosorbent assay for serum using an animal diagnostic kit typically consists of three main parts. We will use the example of classical swine fever ELISA kit for explanation. Part 1. Preparation of serum samples. The preparation involves the process of blood collection and centrifugation to obtain serum. Typically, for blood collection, we use a vacuum blood collection tube or a disposable veterinary blood collector. The amount of blood drawn is usually around 2 to 5 milliliters. Part 2. Reagent preparation and sample preparation. This step primarily involves the process of diluting the sample and concentrated wash buffer according to the instructions provided in the manual. Part 3. Detection, with specific operations following the instructions provided in the manual. The following is an explanation of the enzyme-linked immunosorbent assay procedure. Friendly reminder. 1. Prior to conducting the assay, it is essential to prepare the serum samples, working wash buffer, and sample diluent in advance. 2. The kit should be taken out of the refrigerator at least 30 minutes in advance to ensure that the surface temperature of all components of the kit matches the room temperature, approximately 25 degrees Celsius, thereby reducing errors caused by environmental variations. 3. After separate the required microplate strips, promptly seal the remaining strips and place them back in the refrigerator along with the empty box for storage. Next, we will proceed step by step with the enzyme-linked immunosorbent assay. Step 1. Allow it to reach room temperature. Adjust the room temperature to around 25 degrees Celsius. Wait for it to stabilize, and remove the test kit and the test samples from the refrigerator. In the lab, wait for at least 30 minutes on the bench until the reagents and test samples in the kit reach room temperature before proceeding to the next step of the experiment. After the kit has reached room temperature, remove the individual components of the kit with their labels facing towards you for easy access. Open the required number of enzyme-labeled wells, and seal the remaining wells in the sealed bag. Place this bag inside the foil bag along with the desiccant. Then, put the foil bag back into the empty box, seal the box using a waterproof bag, and return them to the refrigerator immediately at 2 to 8 degrees Celsius. Step 2. Plate setup. This is the process of numbering the wells in advance, including negative control wells, positive control wells, samples, and other components placed in the microtiter plate. Following the instructions in the manual, commence the plate layout by assigning the positions for the negative control, positive control, and the samples on the plate. Typically, the negative and positive control wells are arranged first, followed by the placement of sample wells. 2 parallel controls, one positive and one negative, are set up, each in 2 separate wells. One well is sufficient for the sample. Arrange them in sequential order and maintain thorough documentation. The purpose of setting up duplicate wells for negative and positive controls is to promptly detect and rectify any operational errors. Parallel wells should be set in adjacent positions, either in rows or columns. It is recommended to arrange the microplate in a vertical column format. The following is a demonstration of arranging them in a horizontal row. Step 3. Addition reaction. As per the instructions, the reaction involves adding the corresponding reagents into pre-numbered wells in the specified order. It can be said that an enzyme-linked immunosorbent assay is composed of the process of adding various reagents, making this step crucial to perform correctly. Repeated practice is essential to become familiar with this process. The key points for this operation are as follows. 1. Begin by adjusting the micropipette to the specified scale. 2. When picking up reagents, be sure to check if the labels are accurate. Open the bottle cap with the opening facing upwards to avoid contamination. 3. When using a micropipette, ensure that the pipette tip is inserted below the liquid surface in the reagent bottle to prevent the aspiration of air bubbles or inadequate liquid volume. 4. During the liquid dispensing process, if your hand shakes, you can use another hand to gently support the connection between the pipette and the tip with your finger. When dispensing reagents into the wells, do so swiftly and avoid touching the bottom of the wells or the well walls. It's best to dispense the liquid above the wells and avoid pipetting up and down after adding the liquid. 5. After adding each reagent, remember to change the pipette tip to avoid cross-contamination. You don't need to change the tip for the same reagent. We will now proceed with the addition reaction demonstration. First, dilute the sample, and open the dilution plate. Dilute the test serum 40-fold with sample diluent. For instance, add 5 microliters of serum to 195 microliters of the sample diluent. Adjust the micropipette to 195 microliter. Attach a pipette tip and dispense the sample diluent into the wells. Then switch to a smaller volume range micropipette. Set it to 5 microliters. Attach another tip, then add the sample. And repeatedly pipette up and down for thorough mixing. Change the pipette tips and add each sample one by one. Remove the microtiter plate and add the negative and positive controls as well as the diluted samples into the designated wells. Adjust the pipette to the specified volume mark. Pick up the positive control and gently shake it. Unscrew the bottle cap and aspirate the liquid. Locate the corresponding wells on the microtiter plate and dispense the liquid. If using the same reagent, there is no need to change the pipette tip. After adding the positive control, change the pipette tip, and then add the negative control. Following that, proceed to add the diluted samples. After adding the components as listed in the manual, proceed to incubate as instructed. Incubation. Incubation is the process in which the components added earlier interact with the antigens or antibodies coded on the wells of the microtiter plate. This process requires a specific temperature and should be conducted under conditions protected from light. You can choose from incubation equipment such as a constant temperature incubator or a water bath. As long as the temperature requirements are met, you can choose either one. Ensure the incubation equipment is preset to the correct temperature as per the instructions, and cover any observation window with opaque material to prevent light exposure. Operational demonstration. After adding the negative and positive controls, as well as the samples, cut the adhesive membrane according to the desired number of wells, peel off the adhesive side and place it firmly onto the wells with the added liquids. Ensuring a tight seal, then place the plate on a microplate shaker and gently shake to mix thoroughly. Open the preset incubation equipment, place the plate inside, cover it, and start the timer. Washing. The purpose of washing is to remove unreacted, free components, making washing a crucial step for the success of the reaction. During the washing process, please pay attention to the following 3 points. 1. When dumping liquid from the wells, be sure to use your fingers to support the microtiter plate to prevent it from detaching from the plate. 2. Ensure wells are fully filled with the working wash buffer. Consider using a multi-channel pipette for faster filling and better washing efficiency. Alternatively, an automated plate washer can ensure thorough washing. 3. Drying is done as the final step after the last liquid dumping, and it's not necessary after every step. The drying material, such as blotting paper or a lint-free cloth, must be clean and free of paper debris. After the reaction, remove the plate from the incubator, gently lift the adhesive membrane, support the wells with your fingers, and discard the reaction liquid into a designated waste container to prevent contamination. Once the liquid has been discarded, add pre-prepared wash buffer into each well, 350 microliters per well, ensuring that the wells are fully filled. This step is best performed using a multi-channel pipette or a dedicated plate washer to avoid prolonged delays that may affect the washing efficiency. After allowing it to stand for 30 seconds, discard liquid in the wells, and then repeat the previous steps for a total of 5 wash cycles. After the final washing, pat dry with absorbent paper or a lint-free cloth. then add 100 microliters of the enzyme conjugate to each well. Start by setting the pipette volume to 100 microliters. Open the reagent bottle and attach the pipette tip. Afterward, cover the plate with the adhesive membrane. Open the preset incubation equipment, place the plate inside, close the lid, and start the timer. After the reaction, remove the plate from the incubator, gently lift the adhesive membrane, support the wells with your fingers, and flick the reaction liquid into a designated waste container to prevent contamination. After discarding the liquid from the wells, repeat the washing steps as before, adding 350 microliters of the wash buffer per well, filling each well, and standing for 30 seconds before flicking out the liquid. Repeat this process 5 times in total. Hold the wells with your fingers while discarding the wash buffer. After the final wash, pat dry with absorbent paper or a lint-free cloth. Color development. Adding the substrate reagents for color development serves the purpose of allowing the instrument to recognize and read the results of the reaction through color changes. When adding the substrate reagents, please pay attention to 2 points. 1. The order of adding substrate reagent and substrate reagent B must not be reversed. 2. If you notice that the blue color is relatively light when the reaction time is up, you can consider extending it by an additional 5 minutes. Presentation. Adjust the pipette. Gently shake the substrate reagent A. Unscrew the bottle cap. Attach the pipette tip. Aspirate the liquid and add it to each well one by one. Change the pipette tip. Add substrate reagent B in the same manner as substrate reagent A, step by step. After adding the reagents, cover the plate with the adhesive membrane. Gently shake to mix thoroughly. Place the microtiter plate into the preset incubation device. Incubate for 15 minutes. Add the stop solution. Here, please pay special attention to 2 points. 1. The stop solution is slightly acidic, so avoid skin contact. 2. After adding the stop solution, readings must be completed within 10 minutes to avoid affecting the experimental data. Adjust the pipette to a volume of 50 microliters and pick up the stop solution. Gently shake to mix thoroughly, unscrew the bottle cap, and attach the pipette tip. Aspirate the liquid and add it to each well one by one. Perform the ELISA reading immediately after adding the stop solution. ELISA reading. The ELISA reading is the process of presenting the entire test results through data interpretation. When taking readings, 3 points should be noted. 1. The microplate reader should be powered on 30 minutes in advance, and the wavelengths should be configured, with the primary wavelength set to 450 nanometers and the secondary wavelength set to 630 nanometers. If the microplate reader has only a single wavelength, select 450 nanometers for measurements. 2. The ELISA reading should be performed immediately after adding the stop solution. 3. After completing the reading, The reaction solution should be disposed of in a designated waste container to prevent environmental contamination. Opening the microplate reader, inserting the microtiter plate in the correct orientation into the plate chamber, and then initiating the reading process. After completing the ELISA reading, the data can be exported and used for result analysis. For more information on data analysis, please watch our data analysis video. This episode of the video concludes here. Thank you for watching.